In this video, I will show you an overview of lifestyle imaging workflow on Mica. Please see part 2, 3, and 4 of this tutorial series for more details. Mica is turned on, followed by LASX Mica software. My goal is to image 2x1 tile in every selected well with a 60 micron thick Z-stack. I'm going to start by entering my project name and sample name. Then I'm selecting wall plate template and the plate bottom material. I'm going to image only half of the plate today, so I'm selecting rows E to H. I expect movement in my cells, so I'm selecting dynamic and living. Incubator is turned on by selectively turning on temperature, CO2, and humidity. Temperature must be turned on before other selections. Now I'm selecting the dyes in my assay. I can also enter each fluorophore structure information. My cells are in buffer, so I'm selecting water from my mounting media. All set. I'm clicking Overview Sample. Overview is created. I'm clicking Confirm and go live and acquire. I'm going to image both bright field and fluorescence. I'm checking bright field image quality by clicking live on TL sequence. I'm clicking One Touch for illumination setting to optimize. Next, I'm checking the quality of fluorescence sequence. One Touch is automatically running. I have lots of signals, so I'm reducing exposure time. This will reduce overall acquisition time. I changed a parameter, so clicking One Touch once more. I'm turning off display of other channels, and I'm changing pseudo color to manually find the focal point more easily. I'm adjusting Z position to find the focal plane that I'm happy with. Now I'm going to set up tile scan. I'm clicking wall coverage to set up 2x1 tile in every selected well. All tiles are set up. Next, I'm setting up my focus. Because my cells are moving, I need an adaptive focus strategy to find the focal plane in every well. I am selecting image-based and every sixth position. For more details about focus strategy, please check out part 2 and 3 of this tutorial series. Next, I am setting up Z-stack. I'm clicking 3D imaging and entering Z-stack size 60 micrometer. I only want 13 steps, so I'm entering it here manually. Please note that small volume computational clearing or large volume computational clearing Thunder methods require optimized sampling settings. If you plan to use 3D Thunder processing, please keep the optimized setting. I'm activating both TL and Fluo. Now I'm ready to start acquisition, so I'm going to click Start. Acquisition is complete. To view the images, I'm clicking Results and click through the file names. For more details of each functionality, please check out part 2, 3, and 4. If you have more questions, please contact your local Leica specialist.
If you have any questions or want to learn more, please reach out to us via the link in the description or scan the QR code on your screen. Thank you.